Okay, thank you very much. And thank you for the organizers for having me here. I'm actually very excited to be in this panel of system virologists. For the first time, I don't have my slide of why you should care about single cells. My lab is a new lab. We've been around for 3 years at the University of California in Irvine, and we study HSV1. This is a lab. This is vision. This is a lab vision. We would like to understand the determinants of viral infection at the single cell level and hopefully use this to develop novel antiviral therapies. And our main model system is herpes simplex virus 1. When you think about possible outcomes of viral infection or inoculation or the physical interaction of a virion with the host cell, you can think of at least 3 different infection outcomes where the virus is stopped before it has a chance to express a genetic program, where it gets to where it needs to. In the case of herpes viruses, that's the nucleus, start to express viral genes that never produce progeny, and it can be a productive infection producing progeny, which by itself is a very heterogeneous phenotype. Here you can see our measurements of progeny production from individual human keratinocytes, ranging from 3 to 600 PFUs per infected cell. And my lab would like to know what determines these different infection outcomes on both the viral and host sides. But we're mainly focusing on the host side, both because I'm a cell biologist by training, and the techniques are just much more applicable to hosts than these huge herpes virus, 150,000 base pairs, 80% GC sequences. It's not influenza. So a quick introduction to the first DNA virus of this session, herpes simplex virus 1. It's a very widespread human pathogen. Most of us have been exposed and are infected. Once you're infected, you remain infected. It remains dormant in the peripheral nervous system with occasional reactivation throughout life. Mostly these are asymptomatic, so we carry this virus around, we shed it, we infect other people without ever knowing we do so. In about 20% of cases, we have these recurrent cold sores in the face, mouth, and genitals. And more rarely, and especially in immunocompromised patients, we have more severe manifestations such as keratitis, encephalitis, and neonatal herpes. There is no vaccine and no cure. We have some antiviral drugs that can suppress lytic infection in the epithelium, but they don't get rid of the latent reservoir. In the lytic infection, which is happening in the epithelium, which is what we study in the lab, there are 3 waves of viral gene expression. We have immediate early genes that are expressed as soon as the viral genome enters the nucleus. There's 5 of these. They sort of turn on all the viral gene transcription and shut off a lot of the immune responses. You only need to know one of them, called infected cell protein 4, ICP4, and we're going to use it as a marker of cells becoming infected. Then we have early genes, which are mainly the ones that are going to turn on viral DNA replication. And once viral DNA replication starts, we have a shift from early to late gene expression. And these are all the structural proteins that are going to form new virion particles. And you just need to know one of them, the minor capsid protein VP26, that we're going to use as a marker for the cells having gone through viral, or at least started viral DNA replication. So we're interested in these abortively infected cells that initiate viral gene expression and then stop for whatever reason. So we created this reporter virus that has an immediate early protein fused to a YFP and a late protein fused to RFP. And with that, you can see these abortively infected cells shown here with the arrow, that they turn on the early gene expression, but they never go on to the late genes. With this virus, we can actually quantify how common these abortive infections are. We can infect whatever cells we'd like, sort out individual infected cells based on different viral protein markers into 96 well plates that have monolayers of viro cells. So we can come back to these plates a few days later, and we can easily classify this one cell that we dropped into the well, whether it was producing progeny or not. So depending on the cell type and the multiplicity of infection, it ranges from somewhere between 10 and 30% of the infected cells. And the main take home here, this is not rare. This is not 1 in 10,000 cells that is not producing progeny. All right, so fairly commonly, you have cells that are infected with a virus. And I should point out, these are measurements of cells that express the early protein. So we know the virus got into these cells. It turned on its genetic program, but then it didn't produce progeny. We have a problem with actually studying these, and that's because here are the kinetics of early and late gene expression. And you can see very nicely the primary and secondary infections with the early protein, but the late protein does whatever it wants to in each individual cell. So by the time you have secondary infection starting, most of the primary infected cells haven't gotten to late gene expression yet. So there really is no one time point we can just sort out cells and say these are abortive, these are not, these are secondary infections. We use a genetic trick. We've taken a virus that lacks glycoprotein H, which is part of the fusion machinery. The virus needs that to get into cells. So you grow it in a complementing cell line, but this is a single round virus. You can see that no matter the multiplicity of infection, you have one peak of early, one peak of late gene expression. And by 16 hours, all the cells that are going to express the late genes have done so. So we can fairly easily classify them at that time point for the different infection outcomes. And this is what it actually looks like. So these are uninfected cells. Here on the x-axis is the early protein, and on the y, the late. Uninfected cells don't express viral proteins. That's good. When you infect them with this single round replicate, sorry, you can tell I'm nervous for this crowd. I'm not usually nervous when I give talks. When you infect them with this non-spreading virus and wait 16 hours, you see 4 different infection outcomes. We have cells that don't express any of the viral proteins. We have cells that only express the immediate early ones. We have cells that have some late gene expression, but this is a log scale, so these have 10 times more of this late protein. And this is the only one that are actually producing progeny, as I'll show you. We can take out these 4 different subpopulations by FACS and do all kinds of measurements. We can look at viral DNA replication, and as you expect, cells that don't express viral protein don't replicate the viral genomes. Those that only express the early proteins don't replicate the viral genome. Here you have some viral DNA replication, and here you have more. And when we check for actual progeny production, these here are the only ones that are producing progeny. So all of these are abortively infected cells. Some people call these bystander cells. I'm happy to mince words during the discussion. But clearly they have been exposed to the virus and are not producing progeny. And what this tells us is that during this schematic view of the viral life cycle, there are actually 3 different fail points where cells can fail before they express immediate early genes, before they replicate the viral DNA, and once they start replicating the viral DNA, but not go on to full late gene expression. And we'd like to know why. We still don't know why, and you won't get the answer from me now, hopefully in a few years. We can take these out, and we can do RNA sequencing of this different subpopulation. These are all the viral genes. As I said, herpes viruses have a lot. We have more than 100. This is sort of what you would expect. The one that, the more early on in the process that you stop, the more skewed the viral gene expression to the early genes. What's interesting in these host genes, we have this host shutoff that is very well known for herpes viruses and other viruses. All of these host genes that are usually on get turned off. They actually get turned off even in these abortively infected cells. We have this group of host genes that are only activated in the productively infected cells. And we have quite a large group of genes, about 500 host genes, that are upregulated in these abortively infected cells. There's actually both restriction factors and dependency factors in it, and this is mainly what my lab does these days. We're trying to figure out which of these are restriction factors, which are these dependency factors, which of these have no consequences for the viral life cycle, at least in our cell cultures. And I'm going to tell you 2 vignettes about a restriction factor and a dependency factor. So John did an amazing job introducing interferons, so I don't need to do that. But the first thing we looked at are interferons. And out of the 20-something human interferon genes, only 2 are expressed or are upregulated during HSV-1 infections. Interferon beta, which is a type 1 interferon, and interferon lambda 1, which is a type 3. And quite weird, they're coming from different subpopulations. So interferon beta is activated in these very early abortive infections or bystander cells that don't express any of the viral genes. And interferon lambda is coming from both of them, but mainly from the one that express the early genes and then stop. We have no idea why. We're very interested to figure this out, but we think this is very cool, and that sort of led us to think of interferons and ISGs in the case of HSV-1. I want to point out that interferons are the consequences of abortive infection and are not the cause, and I will die on this hill and fight me later. Okay. There's a lot of known stuff about the interferons in HSV-1, but one key thing to know is that Helen Lazier's group have done the knockouts of the interferon alpha and lambda receptors in mice, and you see different pathogenesis. When you knock out type 1 interferons, the virus goes into the brain and kills the mice. When you knock out type 3, there's more severe skin lesions, but it doesn't go into the brain. So clearly they have different roles in pathogenesis. And we'd like to know which ISGs mediate the antiviral effect against HSV-1 in the human keratinocytes. I swear I didn't put this because John is here. I have this in my deck slide usually. There's 2 ways that we think about how these ISG works, either the death by 1,000 cuts, that all of these hundreds of ISGs have some minor effect for all viruses, Or maybe there's a limited set that is important for each individual virus. So to find that out in our human keratinocytes, we've done dose responses for interferon beta and lambda. Here I'm showing you the % of late gene expression with this non-spreading virus. Lambda is much weaker, we know that, but we can find concentration where they do the same inhibition. So on IC95, about 95% inhibition. And then we can do the transcriptomic, where we just treat cells with interferon beta and lambda at this concentration and see which genes come up. This is 24 hours post-treatment. You can see that for the most part, they activate the same interferon-stimulated genes. All the ones in red are shared. The blue ones are only significant in beta. The green one only significant in lambda, but they're sort of lie up on the diagonal anyway. We can look at the same plot and just notate the known HSV1 restriction factors. There's a bunch of them that are not ISGs. These are constitutively active restriction factors. There's a lot that are weakly induced by these interferons. And there's one clear outlier, which is MX2, which is induced by about 10-fold upon treatment with both interferon beta and lambda. And it is known to have an important role in inhibiting all herpes viruses. So we wanted to look at this a bit more. We've knocked out MXB in our human keratinocytes. These are 3 different monoclonal lines. We've sequenced them. They have different deletions, but they all lack MXB. And with this, we can actually quantify what is the importance of this single ISGs, MXB, to the antiviral effect against HSV1. Here I'm showing you the expression of the early genes, but we've done it for the late as well. This is for interferon beta, and we can attribute about a quarter of the activity of interferon beta antiviral activity to this one ISG, MXB. And it's even higher for interferon lambda. It's about 37%, so a third or something like that. So this supports this limited set model of ISGs. And quite interestingly, even though that interferon beta and lambda upregulate MXB to the same extent, it contributes differentially to their antiviral activity against HSV1. I'm running out of time, so I'm going to skip this quickly, but I love these pictures. We do it in human skin organoids as well. We can make them from our knockout keratinocytes, and we see basically the same. So you see what we treat the regular keratinocyte with interferon beta, we have about 3 log reduction in HSV1, but when we do the same with keratinocyte knockout for MXB, we have about 1 log reduction. So clearly MXB is really important, but it's not the only one. And what is an open question, and we're very interested to find out is which other ISGs mediate the remaining activity of these 2 interferon against HSV1 in the human skin. Okay, the last piece of data I'm going to show you is about a dependency factor, or a presumed dependency factor. This is, again, from the RNA sequencing, and this is snail. It's a master regulator of epithelial to mesenchymal transition. It's not expressed in uninfected keratinocytes. It's not expressed in these very early abortive cells, but it goes through the roof in these early abortive infections, the cells that express the early protein and not the late, and then it goes down in the higher groups. And the reason we looked at this is that when you look at the cells, this is a monolayer of keratinocytes and a plaque forming with our fluorescent virus. You can see the very nice epithelial shape of the uninfected cells, but the infected one looks like fibroblasts. It looks like they're going through EMT. We've knocked out snail. We looked at the infection, and we see a reduction in HSV1 infection. It's very preliminary. Don't quote me on that. We're still trying to make sure this is real, but we think this is a dependency factor, and there's reason to believe from CMV that EMT is important for herpes viruses infection. So snail is activating these abortively infected cells, which really tells us it's activated in the early part of the life cycle, and we're just capturing it in these abortively infected cells because they don't turn off the early program to switch to the late one. With that, I'd like to conclude, and hopefully I've convinced you that abortive infections are common, and HSV1 infected cells often fail to produce progeny, that there are at least 3 different abortively infected subpopulations. Each has its own unique transcriptional program, and in this transcriptomic, we can find both restriction factors and dependency factors, that the type 1 and 3 interferons are inducing distinct subsets of abortively infected cells, and MXB mediate a significant but different part of the 2 interferon responses. There's dependency factors such as snail and DUX4 we didn't talk about today, that's published, that accumulate in these abortively infected cells, and they're really only transiently activated during infection, but we can capture them in these abortively infected cells. And we'd like to know what are these host pathways that are able to arrest infection after it starts, how do they work, how are they triggered, and what is the physiological relevance. With that, I'd like to thank my lab, collaborators, organizers, funders, well, they still fund us. Thank you for listening. This is all very preliminary. Please email me with any feedback or find me later, and I'm happy to take any questions.